All right, physiology students, wanted to get this information to you regarding the kind of just a little overview of the immune system that we don't have time to get to for class on the first day. So so it starts here, just kind of looking at the two general arms of the immune system. So we have the innate and the adaptive, right? So the innate immune system is going to be nonspecific. It's going to have the same response to every invader, right? So it attacks all foreign invaders. It doesn't have a memory. It doesn't get stronger when you've seen that same invader more than once. It just has the same response. This is where we get fever and inflammation from. And it's going to use some of these general kind of tools to destroy things. And then the adaptive side is going to have very specific defenses for each invader. And we need multiple exposures to really build the defenses. So the first time something is attacked by the adaptive system, the response isn't going to be very strong, but it will get stronger after that, and you should be able to build up immunity against that. This is the basis of vaccines and things like that. It's going to use cytotoxic T cells and B cells that make antibodies. This is known as cellular mediated immunity and humoral immunity. So let's look at some of the cells of the immune system. Okay, so first of all, we've got lymphocytes. Okay, so lymphocytes include B cells, T cells, as well as natural killer cells or NK cells. Okay, we find them in the lymph nodes for the most part. Okay, and these are the cells that are really, at least these two are going to be the ones carrying out adaptive immunity, and the natural killer cells are part of the innate system. The MHC1 and MHC2, these are major histocompatibility complex molecules. And what these do is MHC1 is the one that all cells have, except for red blood cells. And MHC2 is our immune cells. And kind of the best way to think about this is thinking about this in terms of kind of an ID card for the cell, right? So kind of proof to show that that cell is supposed to be there. That's what MHC1 is. This is kind of like a general ID card, whereas MHC2 is a little bit more like a law enforcement ID card. Okay, and we'll talk a little bit more about those later. Okay, T cells or T lymphocytes, these are a part of the adaptive system. Okay, their cell life cycles are produced in the bone marrow, but then they mature in the thymus, and that's where they get their name, the T cells. And then they're sent to the bloodstream or the lymphatic tissue. Their job is to help recognize an antigen in combination with that MHC1 protein, and then that's going to activate the immune response. And so specifically, helper T cells are very important for activating the immune response. And then their job is going to be to activate others. Then some of those that get activated are called cytotoxic T cells or killer T cells. Those are going to go and destroy whatever it is that needs to be destroyed. There's also regulatory T cells and memory T cells. B cells are also a part of the adaptive system. They're made in the bone marrow, just like all blood cells, but then they mature also in the bone marrow. That's where they get their B for the B cells. Okay, their main function is to make antibodies. And interesting enough, each only produces one type of antibody. So meaning that if you've been exposed to 1 million different viruses, that means you have 1 million different specific memory B cells that can produce a specific antibody for that virus, for each of those viruses. The term plasma cell is just used for an activated B cell. So this is a B cell that's sent out into the bloodstream, and it's making antibodies and dumping them out, just like this one right here. And memory B cells are going to help remember that invader and be ready to quickly build antibodies next time that invader comes in. So how does innate immunity get adapted? Get activated? There's a couple major kind of receptors that help out with this. The toll-like receptors are one class of receptors that are very helpful for this. This provides the primary stimulus for the cell's innate response to invaders. And there's a bunch of different types, TLR1, 2, many, many different types. And these are kind of specialized for different types of invaders. Okay, and there's also NOD proteins. NOD proteins are there to, these are intracellular receptors, so kind of like the toll-like receptors, but if a invader is able to get into the cell and kind of bypass the toll-like receptors, then we have this other receptor on the inside, these NOD cells, which are able to detect bacteria inside of the cell. And there's dysfunction in these is what's implicated in Crohn's disease. And so both of these, when they're activated, lead to the release of cytokines or activating the complement system, which then will activate the rest of the innate immune system. How do we activate the adaptive response? It's a little bit different here. When we activate the adaptive response, first we need what's called an antigen-presenting cell, or an APC. Antigen-presenting cell, it could be a macrophage, could be a dendritic cell. Those would probably be the two most common that do this. They're going to encounter a foreign invader on the outside, out in the tissues, somewhere out in the blood, and then they're going to engulf that, break it down, and then take that back to the lymphatic system, take it back to the lymph node, where then they're going to interact with a helper T cell. And that is our CD4 cell. So that CD4 cell is our helper T cell. And what we can see here is that we see these antigens came into the macrophage, right? The macrophage has an MHC2, right? So then the macrophage is going to present its antigen, which is shown right here, bound in that MHC2 complex to the helper T cell, and it's going to bind that to the T cell receptor. That's the TCR. The T cell receptor hooks up with the MHC2 complex, holding the antigen, and when that connection happens, along with the release of some cytokines like interleukin-1, that is going to activate that T cell. And then when that T cell becomes activated, it's going to release some more antigens. We see some more here, IL-2 receptor, right? And now that activated T cell is going to cause other T cells to become cytotoxic T cells, which is a CD8 cell. Some of those are also going to become, we're also going to activate B cells. That T cell is going to then activate B cells, and they start producing antibodies. And this cytotoxic T cell and these B cells are designed specifically to look for this antigen only, right? And that is kind of the process there of that activation. Okay, a little bit more about antibodies. Antibodies, or immunoglobulins, there's really 5 main categories of these. And we can see these here, IgG, IgA, M, D, and E. You can kind of see the general actions of those. But when we look at antibodies, we can see that there's really 5 kind of main actions that they do to help destroy invaders. Okay, one of those is neutralization. You can see it kind of can block the active parts of a bacteria or something large like that, so then that can't bind to one of your cells and damage it. We can also have agglutination, which that agglutination causes a bunch of cells that are already infected to stick to each other, which then kind of clumps them together, makes them unable to destroy or cause any problems. We have precipitation, which would be with very small antigens, soluble antigens. If we stick a bunch of those together, then that becomes heavy, and then they kind of fall out of solution, and then they're not able to have a negative effect on your cells. We also have activating the complement system, which leads to cell lysis. Complement system is using plasma proteins that are already floating around in the blood. If we activate those with antibodies, then those proteins can come together, form pores that stick into the side of cells that are damaged, and then we can destroy that cell. And then a couple other things that happen here. This leads to agglutination as well as precipitation, can lead to greater phagocytosis occurring, and then that precipitation also can lead to the inflammatory process. One way to remember this is with the acronym PLAIN-O. Okay, so we have precipitation, we have lysis, we have agglutination, we have neutralization, and then the O is another one that's not on here called opsonization, which really opsonization is kind of just like marking or flagging a cell or a molecule for engulfing by a macrophage or another phagocytic cell. So that opsonization is there. All right. Okay, and then just a couple kind of little review on these different leukocytes here. Okay, so leukocytes are white blood cells. So we've got a bunch here, right? So all these different white blood cells, and kind of the way to look at this is I've got the function there for you. I've got which system it's a part of there. And so what we'll do here is we'll just put letters on these things. And then I would encourage you just to pause this for a second and see if you can fill in the correct answers here. Okay, so the first one there, the most abundant white blood cell usually going to have first contact is our natural killer cell. Or sorry, not our natural killer cell, it's our neutrophil. So I'm going to put C right there. Okay, the second one there that's going to be specialized in virally infected cancerous cells, technically a lymphocyte. This would be those natural killer cells. That is E. Okay, and then we got the next one there that's circulating around, turning into macrophages. This is going to be a monocyte. So these are kind of macrophages that are before they turn into macrophages when they're in the blood. Destroying invaders, cleaning up debris, stimulate the adaptive system. This is an APC, and these are macrophages. Okay, the ones that are going to release histamine are going to be our mast cells. Roam around the body, catch antigens, also an APC. Those are going to be our dendritic cells. Okay, the ones that make antibodies are going to be our B cells. So it's going to be G. The ones that stimulate B cells and T cells, those are going to be our helper T cells. Directly kill specific antigen, part of the adaptive system. That is going to be our cytotoxic T cell. That's going to be D. And then remember specific antigen for next exposure. This would be our memory cells. So that could be memory T cells or memory B cells, but memory T cells, the only one that's on is an option there. And then to help suppress the adaptive system, these would be our regulatory T cells, which would be F. All right. Okay, and then this is just a flow chart that kind of walks us through the process of activating the immune system and activating inflammation. And so I'm going to start with tissue injury here, right? When tissues are injured, that means some cells are damaged, typically. Some other structures are damaged. But the thing we want to think about first is damaging cells. And so when we damage cells, we release some chemicals from the inside of the cell. So cell contents are spilled out into the interstitial space. And those cell contents are going to activate cells around them. And when we activate cells that are still intact by those cell contents, we activate something called nuclear factor kappa beta. And that nuclear factor kappa beta is going to be kind of a key activating process. So we can see a little bit more information on it here. It gets activated inside of intact cells via cytokines. Okay, so cytokines are released from some of the immune cells as well as some of the other cells around the area. They can also be activated by viruses or antioxidants, and that's going to start transcription of many inflammatory mediators that cause the whole inflammatory process. Okay, so we got cell contents as well as cytokines and other chemicals that are leading to activation of the immune system as well as activation of this nuclear factor kappa beta. Okay, now what we see here in red, we see the 4 cardinal signs of inflammation, and then in yellow we see the 3 primary responses to this. Okay, so number 1, we're going to get some vasodilation, right? Histamine is a major vasodilator. That's one of the things that's going to do that. We're going to get increased capillary permeability. Capillary cells or capillaries are going to relax, open up, and that means blood and more content's going to be able to get to that area and get out into the interstitial space. So that allows for fluid and leukocytes to enter the interstitial space. Okay, that's going to help us to clear out toxins and debris, but it's also going to deliver cells and clotting factors. They're going to help with clotting. Okay, that vasodilation is also going to increase blood flow, which leads to some of those things there, but it also leads to the heat and redness that we see as two of the cardinal signs of inflammation. Let's go back over here to pain. Some of those chemicals that are released, some of the interleukins as well as some of the cell contents can directly stimulate pain receptors and lead to pain. We also get chemotaxis, which means other cells, immune cells in particular, migrating towards that area where the damage is, and that those are attracted by the interleukins and other things released. So this is going to be more white blood cells, more phagocytic cells, okay, which is going to contribute to some of the swelling, but also help to release, also help to get there and clean up and deal with what needs to be dealt with. And swelling is also going to be caused by that capillary permeability as well as the increased blood flow. Okay. All right, guys, that is it. We'll leave it there, and we'll see you guys soon.